Hi, I'm Ethan,
And I'm Brian.
and this is Lab 9 Michelson Application Procedures. So for measuring the wavelength of the green mercury light source, what we did is we first aligned the interferometer so that we could see circular fringes. And once we did that, we moved the micrometer slowly until we counted 100 fringes and used that value with our calibration to calculate the wavelength of the green mercury source. The value we got for the green mercury source was 514.2 nanometers. This value was off compared to the actual value mainly due to the error when counting the fringes through here. If we mistakenly counted off by one fringe, we would be off by about 5.19 nanometers if we missed one fringe. To know the value of the wavelength within 0.1 nanometers, we would need to calculate about 5,190 fringes.
So the second part of the experiment was very similar to the first part, but instead of using the mercury source, we used the sodium doublet source. And so by once again counting 100 fringes by rotating mirror 1, we were able to calculate the wavelength of the light source. And we got it to be 553.7 nanometers, which again was off because of the counting error caused by number of fringes moving too fast.
So what we did for the spectral line separation theory is first we aligned the interferometer so that we could see circular fringes in here. And then what we did is we moved this down so it was close to zero as possible. And then we moved the fringe pattern until we saw two nulls and recorded the value of each of those nulls at the micrometers. Then we calculated the wavelength separation, which was approximately 0.531 nanometers, which gave us an 11.5% error when compared to the accepted separation of 0.6 nanometers. But like before, the percent error is due to the use of the experimental data, which was due to human error.
So we calculate the refractive index of the air using this helium-neon laser to produce circular fringes and place a gas cell between the mirror and the beam splitter. We then pumped air using this pumper into the cell as we count the number of fringes that moved as the air was pumped and record our initial and final pressure values. After measuring the length of the cell and the temperature, we got the slope change of refractive index with respect to pressure to be 4.695 times 10 to the negative 6 per centimeter Hg. This value is used to create our plot, and we calculate the refractive index in the room temperature as being 1.000285. The challenge question utilized the slope that we calculated from the last part in order to find how many fringes it would take to get a pressure change from Tucson elevation to Mount Lemmon elevation. And after redoing the calculation backwards, we found that there would be 7 fringe changes for that change in pressure.
Subscribe!